This method can help answer key questions about environmental virology, such as the emerging of new diseases. The main advantage of this method is purifying virions away from cellular components and using those virions to extract DNA for sequencing.
I first came with this idea when we found out that the plants in our greenhouse is infected with one or more unknown viruses.
Individuals new to this method will struggle for 3 reasons. First, homogenization is a critical step to ensure quality yields. Second, differential centrifugation can produce multiple pellets, and you need to know how to identify and separate those pellets. Third, bioanalyzer can also be difficult to interpret.
A laboratory coat and gloves should be worn for all steps of this procedure. Start by cutting 80 to 100 grams of leaves from diseased plants previously frozen at -20 degrees Celsius. Then, grind the leaves in a Waring blender using 200 milliliters of grinding buffer and 0.5% sodium sulfite. Transfer the homogenate to a 1-liter beaker. While still wearing appropriate personal protective equipment, add 18 grams of urea and 25 milliliters of 10% non-ionic detergent to the homogenate and cover the beaker with foil. Stir with a magnetic stirrer overnight. On the following day, transfer the homogenate to 250 milliliter centrifuge rotor bottles and centrifuge in a